Hi, I'm Raghuveer Parthasarathy from the University of Oregon. I'm recording the talk I would have given at the 2020 American Physical Society meeting, which was canceled. This is a slightly modified version of that talk. So I'm going to tell you about some of the things that my lab has been doing, looking at sublethal antibiotics and gut bacteria. So these days, we all know that each of us is an ecosystem composed of one human and many trillions of microbes. Most of these bacteria live in our intestine, and they play important roles in a lot of processes ranging from digestion to immune response to a wide variety of diseases. In general, we have a poor understanding of what controls gut bacterial populations and how things like how perturbations like drugs and changes of diet and so on affect gut bacterial communities. The particular perturbation that we've been looking at are antibiotics. And it's been known for quite a while from sequencing-based studies that antibiotics can induce quite large, long-lasting changes in the composition of gut bacterial communities. And this in itself isn't surprising because antibiotics do target bacteria, but the surprising part is that this is the case even at low, that is sublethal concentrations of antibiotics. And these levels are especially concerned because low antibiotic concentrations are often found as environmental contaminants. So how could this be? How can weak antibiotics have such a large effect on gut bacterial communities? So we decided to look at this and look at it quite literally by actually visualizing the response of gut bacteria to antibiotics. Most of what I'll tell you is actually published in a paper that came out at the end of 2019, and there's some newer things also at the end. We use as a model system Zebrafish for the usual reasons that it's commonly used as a model system. It's a vertebrate. At young ages, it's transparent. So here I'm showing you a larval zebrafish at 6 days post-fertilization, and we put a red dye into the gut here. But also it's amenable to so-called gnotobiotic techniques. So one can create zebrafish to be initially germ-free, that is devoid of gut microbes, and then add in particular species just to the water around them and thereby do controlled experiments. And these especially were techniques developed by our close colleague Karen Gilliman at the University of Oregon. And the particular project I'll be telling you about is a collaboration between her group and mine, led especially by Brandon Schloman, who's a graduate student in my lab, and Travis Wiles, who's a postdoc in the Gilliman lab. So to image bacteria, we make use of a technique called light sheet fluorescence microscopy. And I won't go into the details. You can read about them, for example, in this review paper listed at the bottom. But what it gives us is high-speed, three-dimensional imaging over large fields of view, in fact, the entire zebrafish intestine, with single bacterium resolution. It also has low phototoxicity, so it can image for long periods of time. As a model antibiotic, we make use of ciprofloxacin, which inhibits DNA replication and which is quite commonly used and which is often actually found as an environmental contaminant. So to get at just basic mechanisms of what antibiotics can be doing, we do very simple experiments in which we look just at what happens to one species of gut bacteria. And there's two different mono-associated cases I'm going to tell you about. The first one involves the species of zebrafish native Vibrio species, which is extremely planktonic, and that is individual and motile. So here what we're looking at is just one optical plane in real time within a zebrafish gut, and each of these little specks you see is an individual Vibrio bacterium. And as you can see from the movie, they're very motile. They're zipping around like a swarm of bees within the intestine there. So first, let's look at what Cipro does to this species. Now, even before imaging, we can do an even simpler experiment in which we just take germ-free zebrafish. We allow them to be colonized by this native Vibrio species. After a day, we add in a weak dosage of antibiotics, about 1/10 of the so-called minimum inhibitory concentration. And then a day or two after that, we dissect out the guts and plate them to see what is the population of bacteria that existed within the gut. What we find is that if we look at the concentration in the water, it drops by about a factor of 10 with antibiotics compared to without. The abundance in the fish, though, drops by a factor of about 100, a much bigger effect. So why is that? Well, visualizing what the bacteria are like in the gut, we find something quite dramatic. That swarm of bees has now become very sluggish and, in fact, filamented individuals. And those sluggish filamented bacteria are then easily aggregated, just even by entanglement, and they're easily transported down the gut by the mechanical peristaltic contractions of the intestine. So these bacterial aggregates that are induced by the antibiotics are then expelled. And as I'll come back to later, these are in fact live bacteria that are expelled from the gut. So this is in a sense not too surprising because filamentation and loss of motility are well known from in vitro experiments as being common stress responses of bacteria. But what we're seeing here is that these effects are amplified in the gut due to this coupling of aggregation and transport. So what happens in contrast if you look at a species that's normally already aggregated? And what we guessed going into this is that they're already aggregated, so maybe antibiotics will have relatively little effect. It turned out we were quite wrong. So the species we're looking at here is again, zebrafish native bacterium. This one is called Enterobacter, and it normally exists in very dense three-dimensional aggregates. Again, weak antibiotics drop the concentration of Enterobacter in the water by about a factor of 20, but they drop the concentration in the gut by about a factor of 1,000 relative to the untreated case. So an even larger effect than the other species. How can this be? Well, again, imaging shows us something quite dramatic, which is that normally you have large clusters and small clusters, but with this weak antibiotic, we see an enormous suppression of small clusters. This took us actually to a very general question, which is, suppose you have an aggregating species, how can it persist in the gut given that there's a strong coupling between aggregation and transport out of the gut? Well, nucleation and growth of new aggregates must be crucial to all of this. Can we turn this into a quantitative model? So if you stare at these movies for a while, you'll realize that there's a couple of key processes going on. There's aggregation of clusters, fragmentation, growth, and expulsion. And you can write down a simple kinetic model for all of these things with various parameters in them. And I won't go into this in detail. You can read about this more in the paper, but I'll point out that these aggregation and fragmentation components to the model arise commonly for decades in polymer and colloidal physics models. And from that, we know a lot about how these can lead to phase transitions, how they can allow us to predict cluster sizes, and so on. The growth and expulsion are essentially where the biology is in sort of cell division and transport down the gut. So there's several parameters in these models, but interestingly, we can actually fit these from image data and then predict the cluster size distribution that we should get of bacterial aggregates with no remaining free parameters. So what does this look like? Here I'm showing you the measured cluster size distribution from actual image data of larval zebrafish as the points here. And then in pink, we have the prediction of this kinetic model with no remaining fit parameters, and as mentioned, and these fit stunningly well. Suggesting that this is in fact a very good quantitative framework for thinking about gut bacterial populations. This also suggests a phase diagram for intestinal abundance with an extinct phase in which you shouldn't have any steady state population allowed and a non-zero population. And the untreated Enterobacter exists in this non-extinct phase, but the parameters that we measure for antibiotic treated fish move this bacterial population into the extinction zone of this phase diagram, consistent with what we see of having very low populations upon antibiotic treatment. So one might think this is perhaps just some peculiarity of zebrafish or these bacteria, but we don't think so. Basically because, as mentioned, these responses like filamentation and loss of motility are very general responses of bacteria to stresses. And transport is a very general feature of intestines. And so it suggests that this is in fact a general mechanism for antibiotics to have major effects on the gut microbiome. Now, all of this looks at what's happening within the gut. But as I mentioned, bacteria are expelled from the gut, and these are in fact largely alive. So here we're doing a live dead stain in green and red here on the expelled contents of a antibiotic-treated bacteria. There's a lot of viable bacteria there. In fact, a similar fraction to what you see in untreated fish. So can these antibiotic-treated bacteria colonize new hosts? So to assess this, we've done experiments in which we inoculate fish with either a pink or a blue, as illustrated here, Vibrio Z20 bacteria, and we expose one flask of these, let's say the blue bacteria fish, to antibiotics for some amount of time. Then we put these together into a new flask for, again, some period of time, during which we might have expulsion of bacteria from these hosts. Then we wait and bring into this flask new germ-free fish. The question then is, what colonizes these new fish? The pink bacteria, the untreated ones, the blue ones, the treated ones, or both? Now, there's a wide parameter range to look at, but in the things that we've been examining so far, it certainly appears that the antibiotic-treated bacteria can colonize new hosts. In fact, under most conditions, they do this about as well as the untreated bacteria. Here, what we're looking at is, again, a single optical plane in the intestine, and we see swimming around in real time both the pink and blue bacteria in these recipient fish later on. So there's quite a bit to do to quantify the rates of these transmission and colonization rates, but nonetheless, clearly these antibiotic-treated bacteria can be quite good at colonizing new hosts, and it suggests a mechanism for spreading, in fact, antibiotic resistance, as these bacteria are the ones that have experienced antibiotics in the past. So in conclusion, low-dose antibiotics can have large impacts on gut bacteria by enhancing aggregation, and that, coupling to transport, can lead to large population collapses. These expelled populations are viable and can colonize new hosts. And in general, the physical properties of the bacteria and their environment are really important to the biome. As mentioned, this is work from Brandon Schloman and Travis Wiles especially, with also a technician, Elena Wall, in Karen Gilliman's lab, and it's been a collaboration between our different groups. Oh, I shouldn't cover up the funding. There we go. Thank you very much.